Welcome to this podcast from Neurogastroenterology and Motility. It publishes original research and topical reviews on basic and clinical aspects of gastrointestinal sensation and motility, as well as brain-gut interactions. It is the official journal of the American Neurogastroenterology and Motility Society and the European Society of Neurogastroenterology and Motility.
So welcome everyone to this month's podcast from Neurogastroenterology and Motility. My name is Adam Farmer. I'm a gastroenterologist at the Wingate Institute at Barts in the London School of Medicine in London in the UK. And it's really my great pleasure this month to welcome my colleague and indeed friend, Dr. David Bulmer. David is a senior lecturer at the Wingate Institute and is a recipient of the MRC Skills Gap Fellowship. And his main research interests concern the basic science of visceral pain, both in health and disease. So David, welcome to the podcast and many congratulations to you and your co-authors on your paper entitled Evidence of the Role of GTP Hydroxylase 1 in Visceral Pain. So perhaps we could begin, perhaps you could tell me a little bit about what prompted you to perform the study, particularly around the pain protective haplotypes of GCH1.
Yeah, sure. Well, thanks for asking me to come along. I was thinking there's really sort of three main drivers, I think, for this study. The enzyme GTP cyclohydrolase has been around in the neuropathic pain field for some time, and gene expression studies done a few years ago have always consistently shown that levels of this enzyme are very, very high in neuropathic pain models. So this is actually a little bit different to perhaps what most people consider, because we tend to think about the pain-sensitive ion channels or receptors for things like prostaglandins. So this is actually a slightly different way of looking at pain. And no one's really looked at it in the visceral pain field. This was sort of the first prompt. And the other really interesting thing about this enzyme, as you mentioned, is there is this pain protective haplotype. So in the original studies, and this was work from Clifford Wolf's group at Harvard, they wrote a particularly outstanding Nature Medicine paper in 2006, and they did lots of basic science in that paper, showing a role for GCH1 in neuropathic pain. But they also looked at patients that had pain because they'd undergone some kind of nerve crush injury. So they had large levels of neuropathic pain. But what they found, there was a subgroup of patients with actually quite low levels of pain. And when they looked at their genetics, they found that they had mutations within this GCH1 gene, and actually they were producing lower levels of the enzyme, and they were producing lower levels of the product tetrahydrobiopterin, and this fitted with the lower pain levels. So we were particularly interested for two reasons there. One, that there's this novel pain role for GCH1, and two, there was the ability to actually try and get at it within humans by looking at this genotype, which hadn't really been studied for visceral pain. So we felt these were really good reasons. The third reason, which I won't talk too much about, but there has been some breakthroughs recently as well in terms of drugs which can target this pathway. So it was kind of attractive there in the sense that if we found exciting effects, then I think genuinely there's a real opportunity into the future to actually develop a drug to target this approach.
Yeah, that's really interesting, because I think some of Professor Wolf's work showed a strong association between a sort of pain protective GCH1 haplotype, and this was associated with lower pain scores, more mucin, indeed better outcomes for patients with chronic pain syndromes. So that brings us nicely around to the study that you and your colleagues have performed. What was your hypothesis as you embarked on this project?
Yeah, so we had two hypotheses at the start. So kind of based on the animal data, we felt that this enzyme would play a role in stimulating sensory nerves, so it would cause pain peripherally. But we felt, based on its role in the production of nitric oxide and the production of various monoamines like noradrenaline and 5-HT, that there was possibly a role in central sensitization as well. So going in, we had these two hypotheses. One, it would play an important role peripherally, probably largely in inflammatory visceral pain by activating the pain-sensitive nerves. And in the study, that's the first model we use. So we use an irritant model to produce a local inflammation and then measured behaviors in rodents after that. And then our second hypothesis that it would be contributing to central sensitization, we looked at using a very well-established model that Kazim Aziz has set up and runs at the Wingate, where he looks at central sensitization in response to acidification of the esophagus in healthy volunteers. So we're very lucky that we had both models to look at this. And those were our two hypotheses going into this, a role in peripheral sensitization and a role in central sensitization.
So how did you test that hypothesis then?
Yeah, so the first hypothesis we tested using an inhibitor, a small molecule inhibitor called DAHP, and that blocks GCH1 activity. So we used an irritant test in rodents, so intrarectal mustard oil. These produces behaviors which we were able to block with the inhibitor, establishing that there was an activation of GCH1 acutely following this inflammatory challenge, and we were able to block that with the inhibitor. The second thing we did was look for this pain phenotype, so this pain protective phenotype, within patients Kazim had studied for central sensitization. And our hypothesis going in was that some people sensitize very readily to acidification of the esophagus, and some people don't. So we felt that we would find this genotype actually in those patients that didn't sensitize going into the study, which wasn't what we found when we then looked at the genetics. And actually we found something else. We found that there was an effect, but it wasn't necessarily in that central sensitization, but it was more in the ability of people to adapt or habituate to noxious stimuli.
And then there were the key results from the second study, but what were the key results from your first study?
Well, the first study really was that we had a strong effect, and it's an acute effect. So, if we pre-treat 15 minutes prior to the challenge, we get a very, very strong reduction in pain behaviors. So something's happening after we give the mustard oil, which switches on this enzyme very quickly, and it produces mediators that we haven't looked at yet. I mean, probably nitric oxide, which have a very acute effect, either on the sensory nerves themselves or potentially on immune cells in the vicinity. And there's quite a literature around in terms of GCH1 contributing to nitric oxide-mediated activation of lymphocytes. So from the first study, there's a very clear, immediate, acute role for GCH1 in visceral pain. So in response to a trigger, this enzyme's being switched on somehow, and that's producing mediators downstream which are activating sensory nerves. So there's lots to look at there in the future.
Yeah, so that's quite exciting, because I think there are probably a number of important clinical implications, particularly the findings from Study 1, particularly around at-risk patients. And the groups that spring to mind would be patients who are at risk for post-infectious IBS or indeed for chronic pain, whether that be in post-thoracotomy or post-herniorrhaphy pain syndromes. Are there any other clinical implications that you can maybe touch on?
Yeah, I think so. I think it's that kind of concept of preemptive analgesia, isn't it? So if you're encountering an insult that you think is going to lead to chronic pain, switching off this mechanism is likely to be helpful. Interestingly, very recently there's a paper in Pain, I think this month, showing association with this pain protective haplotype in GCH1 and I think surgical outcomes for hernia repair. So it kind of grows. Yeah, I think very much IBD pain would be my first port of call for this. And certainly one thing I would be interested in is to look at the prevalence of this pain protective haplotype in IBD patients that seem to have relatively low levels of pain despite having quite substantial inflammatory disease. There's definitely, I think, a subset of patients, of IBD patients that don't really have pain, that probably should have more pain than they experience, which might suggest they have a cluster of these pain protective genes as opposed to sort of pain causing genes.
So, what were the limitations of your study?
Well, I think it's really just detail then of mechanism. I think this was that first sort of pilot type study. So we haven't really fleshed out precisely how GCH1 is getting switched on and what it's doing thereafter in terms of the preclinical work. In terms of the clinical study, it's cohort size. We felt we had a nice reproducible model, and the literature tells us that you can see effects of this haplotype in healthy volunteers. So you can provoke pain responses that use it. So we think our rationale was reasonable. But the obvious thing now would be to go into big cohort studies, ideally in IBS patients. And I completely agree, Adam, that post-infectious population would be the one, really. And I'd love to look at the prevalence of the haplotype, in a sizable population of those patients. Difficult study.
This offers a really new and cutting-edge mechanism that may have some therapeutic translation in the future. So with that, David, I'd like to thank you and your co-authors very much for really an excellent paper and for assisting in this month's podcast, and also our listeners for tuning in. I look forward to welcoming you again on another instalment next month.
Further information about this paper can be found on the journal website. We hope that you have enjoyed this podcast, and we look forward to welcoming you to next month's edition.